Mr. Commissioner, Mark East for Government of Canada. At the end of my questions before the break, I neglected to mark the document that I looked at as an exhibit. That's tab 3 of Canada's list of documents, CAN 025061. I'd like to mark that as an exhibit.
We marked as 1052.
Thank you.
Mr. Commissioner, Cliff Prowse for the province of British Columbia. I've assured everyone I will be extraordinarily short this afternoon. My first question, I think I will ask of Mr. Van Aglen in particular, Dr. Van Aglen. And it has a follow-up on my friend's question about money and research and how do we figure out how to spend things. First of all, are you aware of the Canada-wide strategy for the management of municipal wastewater effluent, February 2009?
I'm aware of it, but specifically...
You were not involved in the process? You weren't involved with...
I was not involved.
You were not involved with Mr. Arnott, who will be on the next panel?
No.
One of the recommendations in that is a recommendation that a committee talk about, a federal provincial, I think, committee, give consideration to science and research projects. Are you aware of that recommendation or that committee?
I'm not aware of the committee.
All right. Secondly, Mr. Lunn, so I'm referring, Mr. Commissioner, to Exhibit 14, and if the witnesses might look under question 29. This was the question about the federal, provincial, and local governments joining force to deal with things on the Fraser Basin, and if you look at the box on the right...
Excuse me, would you turn your mic over to the...
I'm sorry. If you look at the box on the right, you'll see reference to the Fraser River Estuary Management Program. Are you aware of that, or have you been involved with that, Dr. Van Aglen?
I'm aware of FREMP, yes.
And is that something you've been involved with?
Indirectly, we would have received some samples from the FREMP program in the lab.
All right, and the Burrard Inlet Environmental Action Plan, are you aware of that?
I'm aware of it, but we don't participate in that study.
And the Fraser River Action Plan, I think you did refer to?
Yes.
And did you participate in that?
Yes.
And we've had reference to I think the Georgia Basin Council?
Georgia Basin Action Plan?
Yes. Were you involved in that?
Yes.
And we also have the Fraser Basin Council. Are you aware of that?
I'm aware of it, but no participation.
All right. Those are my questions, Mr. Commissioner.
Thank you, Mr. Commissioner. For the record, Leadham Initial T, appearing as counsel for the Conservation Coalition. And I should indicate for the record, Mr. Commissioner, I have two of my clients who have been present throughout this morning who have sat at council table with me, and they are Mr. Douglas Chapman. Mr. Chapman is with the Fraser River Keepers. He's a former Crown prosecutor from Ontario and called to the bar in BC's retired now. And I also have Ms. Christiane Williamson, who is with the Georgia Strait Alliance.
Thank you very much.
I want to begin by thanking you for a very interesting discussion from this morning, and I hope to pick up from some of that discussion and carry on. And I want to start our discourse by actually looking at some general topics and then sifting through until we get to wastewater. We doing the work of the Commission generally deal with topics, subject matter, topic by topic. And so if this is Tuesday, then this is wastewater management. And so I'm trying to understand in the context of more ecosystem-based management how wastewater functions or fits into the general Fraser River ecosystem, and whether or not you as scientists should be concerned about loading, about cumulative effects, about other sources of contamination and other issues such as global climate change. And I just want to throw that open as a general question, because we often heard so much about ecosystem-based management and people glibly talk about it, but we've yet to see it really be evidenced in action. So perhaps I can start with you, Dr. Ross, because I found some of your comments to be very interesting this morning. Do you have a reaction to how wastewater fits into this general ecosystem of the Fraser River?
Thank you. I think this morning I did indicate that while wastewater treatment plants represent a single entity that falls fairly readily under the guise of regulatory or scientific assessment and monitoring, when one adds up the 90 that would be found within the Fraser River watershed, as well as countless others in the Strait of Georgia, even within the municipal stream, we start to look at a large number and diversity of point source discharges. My concern in restricting sort of our examination of the problematic of contaminants to wastewater discharges would be indeed the fact that we might ignore some other point sources of concern or some other what we call non-point sources of concern. Ecosystem-based management, I think, would be touted as a means by which we can do a better job to manage human activities as they relate to the environment. And I think in that respect, there's a great deal of sensitivity and awareness about the importance of the environment, protecting the environment, and different components of the environment, such that ecosystem integrity does not fail because of the sum of 1,000 cuts or 1,000 impacts. However, it is a management tool. And personally, and professionally, I struggle a little bit with trying to, prepare the scientific method in support of ecosystem-based management. I think there is a struggle at the interface between science and management that there exists a tension. Sometimes that's by design, because science has a certain paradigm it operates within, and science provides peer-reviewed products through publishing in the international literature, and then it becomes international knowledge. It's delivered to managers, policymakers, etc. So there's a role for scientists to be actively engaged in management or ecosystem-based management, but I think there's also a strong need for independent science, where the scientists have the capacity to explore their own issues of concern and to embark on discovery and to publish that. And by doing so, we'll have peer-reviewed, defensible scientific insight into some of the important issues of our time. Touching on the issue of contaminants, finally, I guess I would argue that it's hard to establish certainty outside of a single chemical mechanistic cause and effect kind of response, and generally that's done in the laboratory. However, with the 23,000 odd chemicals on the Canadian marketplace, of which at least 660 are of concern, we end up with complex mixtures. As an ecotoxicologist, I strive to understand which single chemicals might be problematic, but then I also strive to make sure that my understanding is placed in an ecosystem-based world. And very, very difficult for regulators to assess complex mixtures. It's very, very difficult for the guidelines people, CCME guidelines development people, to deal with complex mixtures. And internationally, science and management have really failed, I think, to adequately prepare a paradigm that would address risk associated with complex mixtures, i.e. the real world. I argue at the end of the day that there is an acceptable paradigm for pursuing the complex mixtures approach, and that is through a weight of evidence. That's a weight of evidence where you rely on the best available evidence from single chemical laboratory-based studies to certain types of catastrophic single chemical incidents in the real world, combined with more sort of ecological approaches to ecotoxicology.
Thank you for that answer. But before I move off to get the views of one of your colleagues, you talked a bit earlier today about the precautionary approach. And I'm wondering, in a world where we don't know enough about the effects, both acute and sublethal effects of some of these emerging chemicals. How we factor in that precautionary approach in terms of what are we doing in terms of the regulation of these compounds, these chemicals, how we are letting them into the market to begin with, and how, once they're entered into the market, how we can deal with them, particularly in the case of persistent organico-chemicals.
A difficult question to answer succinctly. A precautionary approach would be, is described in Canadian law, in CEPA and other pieces of legislation, in order to prevent sort of undue harm or impact on the environment, and often in the absence of knowledge or sufficient knowledge to support a certain approach. My experience, I've spent about 8 years in Europe. My experience is the precautionary approach is more protected in legislation in the European community. In fact, they really refer to it as the precautionary principle because it carries more weight in courts of law. In Canada, I think we're somewhere between the American model and the European model. But the precautionary approach, as I understand it, is not really applied. It's more of a preamble to legislation. I think at the end of the day, the precautionary approach reflects societal values. How acceptable would it be to release chemicals that have been shown to be endocrine disrupting and have been shown to cause adverse impacts on salmonids or fish elsewhere? How precautionary would it be to release persistent bioaccumulative chemicals that we know can get into killer whales, and we'll be waiting until the end of this century before killer whales are protected from PCBs? That's to name one class of chemical. So precautionary approach is a policy term, it's a legal term. It's based on some understanding of the science that's out there, and it's adopted, as I would see, to reduce risk.
Thank you. I'm going to turn to you, Dr. Ashley. I'm going to restate the original question that I had posited to the panel in this way, that you're called upon to give evidence about a specific topic, wastewater, and yet you have to take the realization that the world doesn't operate that way, that we don't just have point sources of wastewater facilities operating at discrete points along the Fraser. We have other sources and point sources of pollution, and we have sources of contamination coming from the air. We have lots of different loadings. How do you factor all of that into giving advice to this commission in terms of what we should do about wastewater?
I would follow what Dr. Ross said, is that there has to be some precautionary principle where you have some sort of advanced early warning radar type, that you're looking at things well in advance of them happening, because by the time they happen, they're often too late or too costly to undo. And so the type of sort of proactive monitoring program would be something that would be a decision that we're going to set up a monitoring network with some interdisciplinary group as to what to monitor, and so it'd be done far enough in advance that it would be like early warning. So, because right now the whole planet's changing with climate change, and so you have that as a backdrop by which everything else is layered on top of it, from hydrology to loss of the pine beetle forest. And so it's becoming increasingly difficult to sort of manage in real time, and you need more lead time. And the only way you can do that is by sort of intelligently figuring out what you need to monitor as far in advance as possible based on literature and best emerging science.
We've heard some evidence from other scientists who have preceded you to the panel about global climate change. And I believe Scott Hinch came and talked about global climate change. And do we know enough about if there's going to be global climate change, which is evidenced by increasing water temperature in the Fraser River? Do we know what effect, if any, that might have upon some of these emergent chemicals or some of these other chemicals that you've been discussing, the endocrine disruptors?
I don't believe so. I think most of the information on temperature increases would be just basically on the physiology of the fish, as Dr. Hinch described, the secondary effects on dealing with chemicals emerging concern. I don't think has been looked at very much.
And I'm not going to neglect you, Mr. Van Agelen. Do you want to weigh in on the general question about loading and how do you deal with a point source such as wastewater, knowing full well that the reality of the situation is that the Fraser River is subjected to a lot of contaminants on a daily, consistent basis?
Dr. Ross summed it up quite nicely, but I'd weighed in on the fact that it's the sum total of the constituents that are going to elicit the toxic response. So while we will be able to regulate under SEPA or the chemical management plan, those chemicals that have been identified through research as being particularly nasty, it's once the mix gets into the effluent that you have all kinds of other activities happening that we may do research on a parent compound, that once it gets into a complex mixture, there's all kinds of other activities that come into the chemical transformation of these products. So I, sticking to my trade, believe that having robust detection techniques or systems in place that we can monitor or predict the outcome of the mixture before they elicit or create effects at the population level. So hanging my hat on the precautionary principle, yes, that's what we should be doing for good, robust science, but keeping the principle that we need warning tools or warning mechanisms to predict and also not only predict but let the regulatory agencies know that there is issues with particular discharges or point source discharges to potentially eliminate effects at the ecosystem level.
Thank you. This commission was called into being basically because of the decline of the sockeye in 2009, which was itself a reflection of a consistent decline over the last decade preceding that one. And I was wondering, from the aspect of the evidence that you've given, I seem to have got the evidence from Dr. Ross, but I want to confirm it again, that you can't rule out that a factor contributing to the decline of the Fraser River sockeye is the presence of contaminants, and specifically contaminants that are located in wastewater.
I don't think we can rule that out, no. Absence of evidence is not evidence of absence, and I think we could get into a discussion about relative risks associated with different practices, but I did point out this morning that in other examples, in other parts of Canada, the United States, and Europe, we've had catastrophic population level consequences associated with single chemicals, some associated with wastewaters, some associated with deliberate application of pesticides.
Right. Dr. Ashley, do you have an opinion on that general question of whether you can rule out contaminants from wastewater as a contributing factor towards the decline of Fraser River sockeye?
I think there's two components to that question. One is relative to 2009, the missing sockeye, and one is relative to the overall stock productivity over the last 30, 40 years. I think the answer to did municipal wastewater effluent have a sort of a smoking gun relationship to what happened to the 2009 return? I don't believe so. Relative to the long sort of decline in stock productivity over the last 30, 40 years, it's been shown that I believe there is a likelihood that wastewater effluent has contributed to that.
And Mr. Van Agelen, do you have an opinion to venture on this topic?
I would follow exactly what Ken mentioned, is that, yeah, it's to specifically target the 2009 decline to one event, most probably difficult to do, but did wastewater contaminants contribute to potentially declines in populations, I would say yes, and echoing what Dr. Ross mentioned about the studies of, in the UK on trout populations downstream of municipal wastewater, showing feminization, dual sex characteristics as a consequence of exposure to xenoestrogens in municipal wastewater. It's a strong likelihood.
I want to now turn to some specific questions, and I want to start with you, Dr. Ross, and see if I can get a document. It's document number 4 from the Conservation Coalition's documents. It should be something that you will recognize readily, Dr. Ross, because I think you're the senior author on this. It's a document entitled Large and Growing Environmental Reservoirs of DECA BDE Present an Emerging Health Risk for Fish and Marine Mammals. Is this a paper that you authored?
Yes, it is. It's actually a preprint. It's available in final published form in the International Journal Marine Pollution Bulletin.
So since this document, you're saying it's now been published in a peer-reviewed journal?
Yes, that's correct.
Might this be marked as the next exhibit, please? And I'm just going to ask you to turn very briefly to page 9 of that particular document, Dr. Ross, under the heading Conclusions. And you go, total PBDE concentrations are increasing rapidly in abiotic and biotic matrices in Canada and have surpassed the structurally related PCBs as the #1 contaminant in sewage, sediments, and water in near urban parts of Canada. Is that true with respect to British Columbia?
Yes, in simple terms, yes.
And specifically with respect to wastewater in the Fraser, would that also be your opinion?
Well, I haven't engaged in any studies of wastewater, but I have colleagues that have been looking at wastewater, and I believe also if one looks at the annual report from Capital Regional District, there is information that would suggest that PBDEs exceed PCBs readily in the effluent from CRD plants, and I would suspect it would be similar for Metro Vancouver.
The next document I wanted to put to you is Commission Council document number 10, please. And this should be an email exchange between yourself, amongst other parties, and a number of other people. And as we go through it, I'm going to ask you, this in part was reflected in what we've now marked, Mr. Commissioner, I believe, as one of the exhibits. I believe it was 1048, the memo to Robin Brown. This is, I believe, a precursor to that. Do I have that right, Dr. Russ?
Mr. Lee, sorry to interrupt. Tab 10 has been marked as 1049. Is there another?
Oh, it has been. Oh, sorry. Thank you. My mistake. If we could take you, if I could just take you through your section of that, Dr. Ross, and I think the easiest way to do that is by scrolling through.
This is the document. Exhibit 1048.
If the highlights in the blue, I think, are yours. Is that correct?
That's correct, and that would come from that same email that was cross-referenced in another exhibit. So, firstly, the first question I wanted to ask you is that this provides to us lawyers a very interesting insight into rulemaking, into regulation, because as I understand it, there is a very compressed timeline in which you are being asked to comment upon proposed regulations. Do I have that right? Yes, we had no, I was not privy to any of the discussions that led to CCME strategy or to the proposed regulations or any of the discussion documents. So, and I would be one of 4 contaminant experts within DFO's Pacific region. And I believe the same would hold for my colleagues as well. So leading up to the email wherein we were requested by our national headquarters to review the proposed or the draft wastewater stream effluent regulations under the Fisheries Act, we had a matter of 2 or 3 days to review all of these regulations, comment, and have that sent back to NHQ.
So, looking at this, at your extract, I want to begin with the paragraph that says, "That said, the proposed regulations lack clarity," bracket, at least for me, on the following scales. And then you talk about the list of endpoints of concern is restricted to BOD matter, suspended solids, residual chlorine, and unionized ammonia. These are with little doubt of concern in a sensitive receiving environment and are cheap and easy to monitor. And you go on to say, however, the only mention of other entities of concern, for example, pharmaceuticals and personal care products, PCBs, PBDEs, E. coli, other pathogens, is in the anticipated benefits of secondary treatment. And you go on to say there appears to be no legal requirement to monitor for these, consider their fate in the receiving environment, or document the way in which treatment will help resolve these issues in either the effluent or the retained sewage sludge. Do you know if your concerns are actually now reflected in the final document that, or is still in draft form?
As I understand it, none of our comments were incorporated into the revised wastewater stream effluent regulations.
You go on to say monitoring is to be carried out by the WWTP operators, that's wastewater treatment plant operators. I don't need to mention the fox and the hen house. So it seems to me that you've got some concerns about the people who are regulating being also the monitors. Is that, or the regulatees being the monitors?
Well, you know, it's an interesting, this was an interesting exercise to me because as a scientist I was asked to carry out a peer review, which is to evaluate, you know, the scientific defensibility of this document or its implications. And of course, as a scientist, I like to have independence, I like to have peer review, I like to have high quality data, I like to be able to access that data in order to review it. So if I make comments that have implications for the management or the application of law, it's because I, as a scientist, view the issue to have relevance in terms of either our understanding of impacts to the environment or understanding of the quality of the effluent. So I think in terms of these proposed regulations, there were some gray areas that made it a little bit unclear to myself how the data would be collected, critically evaluated, that's part of the peer process to me as a scientist, and then acted upon.
Carrying through with some of the other text, if I can, there's a heading, cooperation between the two departments is lacking. There it is. Just before that bold sentence, there's from the Office of the Attorney General 2009, that should read Auditor General's Office. Is that fair?
That's correct.
And then you talk about cooperation between the two departments, and the two departments being the Department of Fisheries and Oceans and the Ministry of the Environment Canada. Is that right?
That's right. This is actually a verbatim excerpt from Sheila Fraser's Auditor General Report of 2009, the chapter on the Fisheries Act, as I recall.
And have you encountered that in your work, where I'm going to call the left-hand right-hand problem, where Canada has two departments and they're not quite connecting? Have you encountered that in your work?
I think when one looks at environmental contaminants, one quickly realizes that jurisdictionally, they're going to be difficult to handle because they involve everything from human health to the health of wildlife in the ocean, on land, potentially the activities of mines and mills and factories and urban centers, many different jurisdictions. And I think it's safe for me to be able to say that I have struggled upon occasion with understanding, first of all, who is responsible for what, and second of all, what I, as a scientist, am expected to carry out in support of the mission of my primary employer, or my sole employer, which is Fisheries and Oceans Canada. I think this Auditor General's report itemizes a major case in point, and that is if one is a scientist working on environmental contaminants and one is worried about the health of salmon or killer whales or shellfish, then one can either deal with the natural resource consequences of that, which would be readily in the purview of Fisheries and Oceans Canada, or deal with single point discharge of effluent from multiple sources, which would be more readily under the purview of Environment Canada and the province.
And I'm going to turn to you, Mr. Van Agelen. Have you noticed in your job that there seems to be some disconnect between Environment Canada and DFO in terms of specifically maybe Section 36.3 of the Fisheries Act?
Certainly not specifically with 36.3, because that's largely the mandate with which the department or the folks that I work with in the department works under. It's pollution, end of the pipe. So our certainly from the perspective of the work that I'm involved in proving non-compliance to 36.3, it's kind of the order of the day for us, with the groups that I work with in Environment Canada. So it's clearly defined as what that is.
So aside from where you've got that interplay where you have to talk to DFO because there's a provision in the Fisheries Act that, as I understand, Environment Canada is mandated to actually take charge of those deleterious deposition provisions under the Fisheries Act. Do I have that right?
That's correct.
All right. Aside from that, is there continual communication between DFO and EC in terms of how you're conducting your business? Are there regular meetings between your managers and things of that nature?
Certainly, in my capacity as lab manager, no. And days gone by, and I would say when I first started with Environment Canada in the early 90s, there was a lot more between our lab and the DFO lab in West Vancouver, which was I guess the home of the pollution control group for DFO. There was a lot more interchange, if you want to call it that, but I would say in the last 10 years, limited to no contact.
I'm going to now turn to you, Dr. Ashley. I don't know if you have any comments on some of the discussion I've been having about the intersection or lack of intersection between the two government departments, whether you have any specific knowledge of that or not. If you have any comments, I'll like to hear them now.
No, I have not been involved in that sort of area for a few years now.
Commission document number 21, which I think is a slideshow or slide deck. Is this something that you prepared, Dr. Ashley?
Yes, I was asked to speak at this summit on Fraser Sockeye by SFU, so I put this together for them.
Right, and so you were called upon specifically to deal with wastewater treatment plants, is that correct?
It was the general effects of contaminants in sewage and their possible effect on Sockeye, yes.
All right, I want to go through some of these with you. And if I can get the third slide in, it should be emerging concerns in wastewater. So you talked there about the endocrine disruptors. We heard some evidence about those, and you identify some of them there. And some of the personal care products. What are POPs?
Persistent organic pollutants.
And then you talk also about nanoparticles, and Dr. Ross talked about those earlier today. Those are emerging concern, are they?
Yes, they are.
And then we carry on, you provide a definition 2 slides after that to what are endocrine disruptors. Is that your definition, or did you get that from the literature somewhere?
That's sort of an accepted literature definition. And I was just, the point there was just to stress at what low concentrations endocrine disruptors can exhibit an effect at, which are lower than the sort of range that typical, more conventional toxic chemistry dealt with at the parts per million or parts per billion concentration.
And you itemize that by saying parts per trillion, 1 second in 30,000 years. That's a graphic illustration of what we're talking about and how minute of a particle that this can be. Is that right?
Yes, most people don't have difficulty relating to a part per million or a part per billion, so when you put it in a time perspective like that, it makes it easier to understand how small these concentrations are.
2 slides down from that, you've got a slide entitled Removal Efficiencies at WWTPs, which I found to be really quite interesting, and it deals with activated sludge versus trickling filter plants. And you say over half of the frequently detected EDCs were reduced by 95% or more by activated sludge plants, whereas 10% of the EDCs were reduced by 95% of the trickling filter plants. So, as I understand your evidence, you said Anasis Island was a trickling filter plant. Is that right?
Anasis and Lulu Island.
And Lulu. So, if we're going to go ahead with some form of secondary treatment for Iona, you would certainly recommend activated sludge over trickling filter. Is that fair?
That's correct.
The next slides talk about what you've already discussed in your evidence: primary treatment, secondary treatment. And then you talk about, I think this is slide 10, effective WWTP design, 2007 study in England. What was this slide meant to depict?
It was along the same lines. This was research that I did while I was working for Metro, because I was concerned about the design of the Anasis plants, and so I obtained some funding and had a literature review done. I did my own, and then had a resident PhD expert who'd written her thesis on this to find out if there were any certain treatment plants that were more or less effective at removing endocrine disruptors and emerging chemicals. And it just so happened at that time in 2007, some of the first major sort of integrative studies were coming out at the same time, one from Europe and then one from North America, and both coming to the same conclusion that trickling filter plants, because of their shorter hydraulic and solids residence time, were less effective at removing endocrine disruptors and emerging chemicals.
And a couple slides after that, I find a slide saying effects on salmon migration, question mark. You can go there, Mr. Lund. Thank you. You say there's something about olfactory imprinting. Could you tell the Commissioner why that is important in the context of wastewater compounds and chemicals?
Yeah, this was a concept that came up as I was researching this, that realizing that when salmon are smolting, they're going through some complex physiological changes where they're both getting prepared for life in a marine environment rather than a freshwater environment, but there's some important developmental changes going on internally in the fish, and the current belief is that during the smolting process that there are sensory olfactory cells are actually being formed at that time. It's almost like a data logger as they're moving downstream, so they're actually growing sensory olfactory cells so they can essentially record what the odor of the stream was as they're moving downstream, so that when they get back on the freshwater phase of their return environment, they'll be able to find their way back to their natal stream. And seeing that on the next slide or two there that shows that the thyroxine is the hormone that's elevated at that time, that triclosan, which is one of these emerging endocrine disruptors, is also a thyroid hormone disruptor. And so this was just basically speculation that this may be one reason that may confound the olfactory imprinting process that takes place during smolt migration, that this type of chemical may be causing some problems with the olfactory imprinting of migrating smolts. I've never read any research on that. It was purely speculative.
All right. But we do know, for example, that all the Fraser River sockeye smolts out-migrate and go past Anasis Island, Lulu Island, and to some extent out to the Strait of Georgia, where they may be subjected to the discharge from Iona. Is that fair to say?
That's correct. As they move down the Fraser River from whatever natal stream, they would be subject to all of the contaminants, whether it be pulp mill or various wastewater treatment plants, all the way down the Fraser, from Prince George, Lytton, Lillooet, Hope, and down to Mission, ultimately to the Fraser Estuary.
And then if we go to the end of the slides, I just wanted to talk very briefly about this ammonia issue that you discussed in your evidence earlier today. And I think if we go 5 slides from the back, we'll get a graph there. This graph is figure 3.1, Anasis Island wastewater treatment plant ammonia concentration versus pH value. What's this graph depicting?
The point was to show that line was the threshold toxicity concentration for ammonia as a function of pH, and just showing where Anasis wastewater treatment plant in an effluent grab was positioned relative to that line. So anything that was presumably to the right of that line would have been acutely or chronically lethal.
And do you remain concerned that this could be a problem for acute lethality in sockeye or any other salmon that's swimming by at the time that you have these low flows?
Yes, I do.
I'm going to turn to you, Mr. Van Agelen, and talk a bit about toxicogenomics. And I think you have Dr. Ashley to thank for your presence here, and I'm going to thank Mr. East for making you available. And maybe by explanation, Dr. Ashley, you were one of the reviewers for the Donald McDonald paper, expert report number 2 to this commission, were you not?
That's correct.
And maybe just turn very briefly, Mr. Lund, to Exhibit 826, pages A11, A13, I think. So it's in the appendices of that exhibit. You start to talk about aquatic toxicological testing, and then you go on to say that Mr. McDonald should take a look at toxogenomics. Is that right, Dr. Ashley?
That's correct.
And if we just look at the slides very briefly, you provide an explanation, basic explanation of what's going on with respect to and maybe you, and with the benefit of Mr. Van Agelen, can walk us through these slides, because I asked Mr. McDonald about it and he was unable to do so.
Well, these are Graham's slides, so I think he should best walk you through it.
Toxicogenomics is basically a catch-all phrase for the application of genomic methods or the genome. The genome is composed, as you see on the slide here, transcriptomics, proteomics, or metabolomics, and that's each representing a different level of organization within the cell, moving from the whole DNA base down to a protein base in the metabolome. What we do, or what I've been doing, is using mostly transcriptomics of expression to determine if there are the fish are eliciting effects as a consequence of exposure. The proteomics and metabolomics are much more in depth and much more precise measure of molecular activity. My lab is not concentrating on proteomics or metabolomics. As I say, we're working on transcriptomics or gene array expression profiles.
If we could just turn to the last slide, because I think that tells us, I think there's one more, Mr. Lund. So that basically describes the approach that you take, is that right, San Agam?
Yes. It's a combination, like I say, what I would say, classical environmental toxicology, combining genomic applications, and then looking at chemical analysis to arrive at a more complete picture of what could be happening as a consequence of exposure.
Now, I was going to do this tomorrow, Mr. Commissioner, with the panel, but I wanted to take advantage of this panel's expertise to see if they can interpret for me some of the data that's contained in reports from Iona Island WWTP effluent. I've shown Mr. East these reports, and I've shown Commission Counsel. They're not on my list of documents, but I intend to put them into evidence to a witness tomorrow. But I want to get one of these witnesses to interpret some of the data that we see here. I'm advised that my client obtained these by going online and essentially getting reports that were available through the internet from monitoring results for operating certificate, sampling location, Iona Island WWTP effluent. And what I want to focus on, looking at the first report, which is dated June 2010 report, is there's various columns, and there's one column entitled 96-hour LC50, and in bracket %V/V close bracket. And then if you follow down that column and look into the row for June the 22nd, 2010, you see there that there's a number 81. Do you have me so far?
Yes.
What is that? Or what does that mean? What does that 81 mean?
Okay, just for the definition at the top of the column,
Yes?
the 96-hour LC50 is defined as over a 4-day period or 96 hours, the lethal concentration that causes 50% mortality to the test population. So in short, Your Honor, there's a series of, how the test is performed is there's a series of cascading concentrations. Usually they'd be 100%, 50%, 25%, et cetera, et cetera. And then over and into that test solution, usually 10 under-yearling trout are added. And then what is, over the course of 96 hours, the analysts would determine the concentration of effluent, which caused 50% of them to die. So if all the fish died in 100% concentration and none of them died in the 50% concentration, the LC50 would be the medium point between the 100 and the 50%. And that is what's been done here to get this value of 81%. That some series of concentrations has been set up where they have calculated that the lethal concentration causing 50% mortality to the test organisms is 81%.
All right. Now, in terms of passing or failing, do you know, can you describe it in terms of, is that a good number, a bad number, or?
Under the Fisheries Act, it's at 100% concentration, greater than 50% of the fish must survive. So this is a failure.
So this is a failure.
Yes.
And if I could just do one more with you, looking at July 2010 report, the next page, once again focusing on the 96-hour LC 50%, looking at the data for July the 14th, 2010, I find the number is 65. Once again, that would be a failure, would it?
That's correct.
Are any of you familiar with the benthic environment that exists below the outfalls, either at Iona or Macaulay Point, Cloverdale Point? And if so, can you offer any comments about the benthic environment and how it may or may not have been modified as a result of the outfall?
have no knowledge of the Macaulay Point outfall, but I've looked at some of the pictures of the transect along the 80-meter contour of the Iona effluent, of which I believe there's 17 monitoring stations, some to the south and some to the north. And so basically what shows up is where the twin outfall pipes discharge, there's a zone of considerable organic enrichment. It's sort of a bell-shaped curve that tapers off north and south as it gets further and further away from the outfall. The main observations was some of the coloring in some of the clams there was sort of a rust color in them, indicating that the sediments had low oxygen concentration, and there was some iron staining in some of the clam shells. There was a different species composition, ones that were more tolerant. The closer you got to the pipe, they were more tolerant of low oxygen conditions. The usual type of effect you'd expect to see in a zone of organic enrichment in a marine outfall.
Dr. Ross, do you have any comments?
I think a couple of things that I would offer up. One would be there are some major differences between Capital Regional District receiving environment and sort of the Strait of Georgia, wherein Iona and Lionsgate in particular empty directly, and that is that from the metro discharges, the plumes enter into an area of high sedimentation. So there's a very big load of fines and particles coming down the Fraser River that would be considered as natural. These fines, these particles will be bearing things over time, and the receiving environment, despite being tidal and having some other currents associated with freshwater discharge from the Fraser, there would be burial over time, and that's of course what I think the natural design of the receiving wastewater system is, and that is we hope that these things will be buried or degraded over time. Capital Regional District, in contrast, is a much more active zone. The Macaulay and Clover Point discharges, they both discharge approximately 60 meters depth, so similar to Iona, but the oceanography is such that it's a very active zone and there's very little sedimentation. So there's a much more limited fingerprint left behind in the local receiving environment, and a lot of the discharge is dispersed, and that would include for these contaminants as well, more dispersed, less of a sedimentation record, less burial, whereas Metro, more burial, more sedimentation. And in both cases, there are reports of obvious there's solids ending up in both environments, and there are some, as Dr. Ashley noted, some effects on benthic community structure. Some alterations have also been noted in CRD annual reports for Macaulay and Clover Points. I think that would encapsulate where I am.
Thank you, Mr. Commissioner. Those are my questions.
Might be a good time for the afternoon break, Mr. Commissioner.
Everyone now recess for 10 minutes?